Good morning, evening, afternoon, or depending on where you are in the world. I'm Mauro Chinappi, and I'm chairman of this session. The first speaker of our session is Aleksandra Radenovic, and she is an associate professor at École Polytechnique Fédérale de Lausanne, and she's going to talk about correlative 3D microscopy of single cells using super resolution and scanning ion-conductance microscopy. So please, Aleksandra, the stage is yours.
Okay, thank you for a nice introduction. I also would like to thank the organizer for inviting me. I also apologize for my camera. I tried everything. I thought that it's a connection with problem, but it seems that it's something inherent. So for today, I would like to actually discuss walk you through the three topics going from solid-state nanopores. So these are the three recent stories from the lab where we use solid-state nanopores, biological pores, and also incorporation of the scanning ion conductance microscopy with fluorescent microscopy. So this workshop focused mostly on ion channels, but there is a large class of nanofluidics artificial devices that are based on a different platform. So solid-state nanopores that are made in silicon nitride or 2D materials. This is one large of the class of the nanofluidics devices where one can study a transport of molecules, fluids, and ionic species at the nanometric scales. So this area of research is, as in ion channels, is motivated by the knowledge gaps in our understanding how single molecules, ions, water, and protons are transported through this small opening. So the platforms that are typically used are nanopores, nanotubes, or recently emerged angstrom scale slits. So in my lab, we focus mostly on the solid-state nanopores and mainly in the pore made either from the 2D materials such as the graphene, hexagon boron nitride, or molybdenum disulfide. So basic flagship applications of these nanopores in the 2D materials are different. So people and also my lab are trying to use this system for DNA, RNA, protein sequencing or detection, taking advantage of atomic thickness of these membranes and as well ability to tune the size of such a pores. Another application of 2D material membranes is in filtration and also in osmotic power generation. For such systems to work, one needs as well to have a very good quality membrane material. So for biological pores, one doesn't need to take care of it. It's biology who takes care of the good quality of the membranes. But as an engineers, here we have to produce these 2D membranes at large scales and also with the high quality. So in 2015, when we started the research in molybdenum disulfide monolayers for the nanopore applications, we have typically dealt with the systems where we had our 2D material grown by a chemical vapor deposition on the sapphire substrates at the ranges of several millimeters. But again, for getting large throughput, reproducibility, and access to many devices, our imminent goal was to translate this type of small scale growth to the wafer scale growth. This is also interesting in the context from the industry because also industry deals typically with devices and applications where larger areas are needed. So for this application, then we started first with the homemade 4-inch wafer scale system, we have designed and on which we could grow this molybdenum disulfide and other transition metal decalcogenides, graphene, and also hexagonal boron nitride at a really high quality, which we tested in and looked at with high resolution using aberration corrected transmission electron microscopes. In aim to produce more and more material, we transitioned from this homemade system in a collaboration with the company that is a leader in a semiconductor. So the Electron, we designed a new system which instead of the hot wall furnace, as in this case, uses a cold wall furnace and allows us to also grow material on a wafer scale with much higher throughput. In parallel to this, we also had to perfect the production of the devices on which we place these two-dimensional, so the support devices on which we place these two-dimensional materials. And for that, we worked hard on the wafer scale production of the devices in using our clean room facilities and exploiting electron beam lithography to define this suspending window in a 20 nanometer thick silicon nitride. So we produce on the wafer scale something like 128 such devices shown here as well. In this image, all of them having a well-defined membrane of a thickness of 20 nanometer of silicon nitride, with the position of the small opening that varies between 80 to 100 nanometers in the center of each of such a membrane. So having developed these systems, we had as well then to transfer a material from our grown system to these substrates, and for that we use two types of transfer. One is transfer based on a PMMA, so using a photoresist on which we coat this wafer on where we grow two-dimensional materials, and then we pick this small area and position a small area of grown material, and we position it directly on top of the membrane. So as you can see here, this triangle indicates the lifted off material from the substrate to the position to the place where we have this opening. Then again, looking with the transmission electron microscope, we can identify the region where we have this 2D material membrane. So this is in this magnification, seeing the membrane is a little bit difficult, but what we see if you focus on my cursor, so we see basically contamination from hydrocarbons or leftover PMMA. So these are these wiggly things, but trust me, below that is a thin membrane of a 3-atom thick material, which is called molybdenum disulfide. Having developed this, it has become eminent that the rate-limiting step in production of our devices is the transfer. And for that, we have also developed a method that allows us to transfer not only one device by one device, but a whole wafer of the system. Next slide. Can you still hear me?
Yes, we can hear you.
Yeah, yeah, I think I will have to stop the presentation because I got stuck. Sorry, it happens.
Okay.
Yeah.
You see your slide, the wafer scale transfer in this moment.
Yes. This is where it got stuck. So yeah, I will continue. So what we want to do is really not to spend the time transferring one device by one device, but we want to transfer the whole wafer of grown material on our substrate wafer. I hope
Okay,
You
now
can now still,
now
yes,
it's
great.
right.
Yes, I'm going back to where I stopped. Okay. Okay, so the system, the whole process is like a following. So again, we start with our sapphire substrate. We put a PDMS. We slowly peel off the 2D material, and we place it on our full wafer, then covering all devices. And then after that, we can use again aberration corrected transmission electron microscope to drill the hole. So I hope you appreciate amount of effort that we have to invest in order just to create a small pores. So while people who are working with biological pores, all this happens naturally, and the nature has basically perfected the fabrication method in which it can make every single of these pores identical. So we are not yet at this stage. So our pores are still terminated stochastically based on how we make them. And in the process of using transmission electron microscope, we have some ability to shape them, but this is still not at the level of the reproducibility that is achieved by the nature. So using this platform, we can then identify single nucleotides, and this has been demonstrated in my lab back in 2015. But what took and what is taking us still a long time is to really transition from the single nucleotide differentiating to real DNA strand sequencing. And on our way towards that application, so we have also examined the different roads, and one is to see if we can see a barcoded DNA. So unlike for the biological pores here, we can basically tune the size of the pore and accommodate the passage of the double-stranded DNA. And in order to create the smallest barcode that was stable, we have used these 3Gs to hybridize to our 3Cs, and in a way that we have positioned it on a 24 nucleotide long piece of single-stranded DNA at the position that is a little bit off-center, in order also to discriminate the entrance of the DNA from 3' 5' or a 5' 3' entry into the system. And this was indeed possible using our technology. So why we are still kind of pursuing this type of research when biological pores are already so great and can sequence reliably DNA. So one of the reason for that is that the density of the devices that we can achieve using this platform can be much higher if we combine the detection of the molecules using a FET sensor combined with the biopore, and this can lead us to a million of devices per centimeter square, unlike what one has in a mean ion device, which is 250 pores per centimeter square. And in this type of detection, what we can do is then detect the in-plane current using the gold, contacting our nanoribbon with the gold electrodes, and simultaneously also measure ionic current as the DNA molecule are passing through the pore. And that, sorry, again, I have to restart. So that is basically principle of the FETs with the nanopores. In which we can, in a parallel, detect both types of signals.
Now you see your screen.
Yes, I know. I will just go to the slide where we stopped. So the geometry of device and the type of device that we that we see. I hope you can see it again.
Yes.
So in the one channel we monitor ionic current, and in the second channel we monitor the current that goes through the FET. So this is a proof of principle devices, and we hope that in the future we will be able to do much more with such systems. So what we need to still work on is what I already mentioned, is reproducible geometry of our two-dimensional nanopores, dealing with inherent defects in the material, scalable growth we have already addressed, and of course using a help and also knowledge from the ion channels, we can, in collaboration with theoreticians, we hope to also translate the knowledge and understanding of the ion transport of the pores when we are approaching the sizes of the hydration layers. So obvious limitation that our pores are having is that we are not having a controlled translocations, and this is something that we are working towards with by developing also other techniques such as this scanning Ion-Conductance microscope that I will address towards the end of my talk. Another application that one could also envisage is hybrid biological solid-state nanopore approaches. So I was very enthused by these FET nanopores, and I initiated a collaboration with the group that was storing an information in artificial polymers. But however, as one can see, we still didn't catch up with the biological pore, so we could not do any decoding of information using our FET nanopore systems. So in order to do that, we have explored so we took one step back, and in a collaboration with our colleagues at EPFL, group of Matteo Del Peraro and Kao Chan, we used a biological platform of aerolysine nanopore to tackle this problem. So I believe everybody is aware of amount of data that we generate daily, and that this is something that will just become more and more difficult to control and as well to store for a long term, especially now in the times of pandemics when also all work is done digitally, and as well we want to not only transmit, but we also want to store this data for a long time. So the process of using the semiconductors has its limitation, although is also progressing. So this DNA, so this storage using the standard digital ways also comes with the high energetic cost. So therefore, people are thinking to use other approaches to store the information, and one of the approaches was to use it at molecular level using a DNA or, for example, proteins. And this has been already nicely demonstrated in the groups of the Church Lab, where they have shown that they can store almost the entire sequence of the movie in the DNA and express it also in a bacteria, and then later read it using the sequencing platforms, also including here biological pores. However, density of the storage in the DNA is super high, but using these artificial polymers, this can be even improved. Not only that, using artificial polymers that are produced by our collaborators at CNRS, group of Professor Lutz, the synthesis of these polymers is also very cheap and very scalable. So when one would compare, for example, the prices of the DNA sequencing and DNA synthesis, we see that the prices of the DNA sequencing went really down, but the prices of synthesis of the DNA are still plateaued. So already what I mentioned is we use this amazing biological pole, which is aerolysine. So this pore, when compared to the other type of other pores like alpha hemolysin, MSPA, or CSG, Nanopore is has the smallest, has the best fitted diameter, so it's highly sensitive, very narrow, and the group of Professor Long has also, where Carl worked before, has shown as well a single base resolution in these systems. So in order to improve this nanopore, many approaches can be done, but the group of Matteo Del Peraro uses approach of molecular dynamics, and they in silico first mutate this pore, and then in our protein facility they express these pores. So they have produced very many mutants of these pores, and we could have tested many of those in order to find an optimal one to read our polymer design. So when we started just with the bare polymers, our signals were not so in the design of the polymers we use N-propyl phosphate as a 0 and 2,2-D-propyl propyl phosphate as a 1. So the sequence of the 5 zeros is depicted here, and the sequence of 00100 unit is depicted here. So what we noticed when we translocate such a polymer that is artificial polymer that it doesn't have any, for example, nucleotides is here. We didn't see any translocation events, likely because there was maybe a difficulty, or either difficulty of the polymer to enter, or they were maybe just as fast that we could not have detected them. So what we designed, motivated by the fact to slow down the translocations, but also to improve readability. So we started to modify this unit polymer with different adding different nucleotides, and we found that the optimal design for our polymer would be if we add 2 A's. So we tested also other types of nucleotides that have been added to the ends. So using this, we could now really nicely discriminate the levels, which would indicate the A's, the zeros, and the ones. And then what we did, we started to shift this position of the 1 along our unit block. So one advantage of this design of the polymer, of having also nucleotides at one end, is that when one wants to also keep these polymers encoding different information in the droplets, for example, using the polymer technology, one uses one important feature of the DNA, and that is that by hybridization, one can easily retrieve a specific piece of information. So this is not possible with these artificial polymers, but by adding just the few nucleotides, we hope that this will become possible. So using this approach, we could then move on to encode not more bits in our system, and as well to proceed towards really encoding an information. So here I'm showing the different designs. So with optimal design, where we have a building block containing nucleotides which are A's, and then our artificial polymer. So seeing that really the highest signal to noise ratio comes with our with this A polymer. So the basic workflow for reading the data is taking the raw data from Orbit Mini, then downsampling, identifying the events, classifying them, and then as well using a deep neural network for assignment of a specific bit. So this has worked quite well up to encoding of the 4-bit, and as I mentioned, as there is not yet any ways of controlling the translocation speed, here is where we start to let's say lose our accuracy, and we hope that in the future we will improve this by adding some sort of a translocation control, and that will allow us to expand this towards more bits that can be encoded in these sequences. So now I would like to basically transition to our third story, which is correlative microscopy using a scanning ion conductance microscopy and one type of a super resolution modality, which is called SOFI. So when we talk about imaging across the biological length scales, optical imaging has been hampered by the diffraction limit, and the nice thing about optical imaging is that it has the ability to monitor cells and molecules in aqueous solution, and also living systems. So electron microscopy obviously has a high resolution, but it cannot operate live. On the other hand, scanning probe microscopy such as AFM and scanning ion conductance microscopy also are compatible with the live cell imaging. So one defining feature of a scanning probe scanning ion conductance microscopy is that it uses a very tiny pipette and monitors the ionic flow through this pipette, and using the feedback, this scanning probe technique never, if the feedback is regulated well, in principle never touches the cell. So it can allow a very long-term measurements and very non-invasive measurements. So it's ideal to be combined with some sort of optical method, because as we know, scanning microscopy types lack the specificity, and combined with the fluorescent probes, we can have the best of the two worlds. So the obtaining a topography from the scanning ion conductance microscopy, and having deeper inside the cells. So scanning ion conductance microscopy has been for a long time around, but what hampered it were two things. So one is the resolution of this type of microscopy, which is defined by the radius of a nanopipette. So the resolution will be given by the 3 radii of the nanopipette, as nicely described by this paper from Tillman-Schaeffer, which examined this on a microfabricated structure. Luckily, in my group, we also did a lot of fabrication, and by also propelling the research in the nanocapillaries, we have found a way of shrinking these nanocapillaries below 10 nanometers by irradiating them in a scanning electron microscopy. So in SEM, we can place very many capillaries and look one by one, and just by electron irradiation that hits the glass locally, we can shrink these pipettes that typically start from the hundreds of the nanometers down to 10 nanometers. Again, this was all nice, but having them reproducibly filled was one of the problems, and which we kind of tackled in a very, I would say, cheap and inexpensive way by using a desiccator that we have placed actually in a microwave, and by applying a short pulses of microwave radiation, we could expel these large and small bubbles from our capillaries. So this was a really enabling step to start to use the scanning ion conductance microscopy with a much higher throughput with the small capillaries, because again, if one has a small bubble, this bubble will rectify the signal, and again, the smaller the pipette is, it is harder to get filled. So to demonstrate really that this is a good good direction. Sorry, again. I don't know what is going on with my program. So we perform scanning ion conductance microscopy measurements using two different diameters of the pipette. So one which was 80 nanometer and one which was the 30 nanometer.
So your screen is again black.
Yes, I know. Yeah, so I open again. Sorry. Yeah, these are the intermezzos between the three stories that we are covering. So now we will have a lot of nice images. So I think the problems are my movies and images that are just too difficult to handle for. Can you see now?
Yes.
Okay, so what we did is take a same cell surface, imaged it first with the 80 nanometer pipette and the 30 nanometer pipette, and one can really appreciate improvement in the resolution. So we can go now even further down, but what we wanted to do first is really to show that the technique are really compatible. So the high-speed scanning ion conductance microscopy and the super resolution SOFI imaging, which basically exploits the blinking dye. So unlike localization microscopy, in stochastic optical fluctuation intensity microscopy, we use information between neighboring pixels, and we look at the correlations of these fluctuations. And by performing a cross-cumulant analysis, we can improve the resolution by looking at the different orders of this cross-cumulant analysis. So the second order is the second cumulant, the fourth order is the fourth cumulant. So that means that that basically we can use this technique also to go beyond the diffraction limit. And as a proof of principle system, we used the COS7 cells, which we labeled with tubulin, looked it in a 3D SOFI, and as well we expressed we labeled an actin in this system in order to also get two color images. In the first realization of this microscopy, we did this on two different setups, and in order to do so, we have to have some ways of navigating through our samples, and for that we have prepared this marked cover slips with this binary system that allows us really to find very many cells, first in the SOFI and then in the scanning ion conductance microscope, and vice versa. So what we can then do is we can do this nice actin imaging, tubulin imaging, and finalize with this scanning ion conductance microscopy, performing the necessary alignment between the two modalities in order to have the two images correlated. We built this nice SECAM SOFI overlay, and that was first done in a 2D geometry, but then we also did it for the 3D. So here again, we see second and third order SOFI. The improvement of the resolution can be appreciated from looking at this ugly white field image, and then doing as well this two color, and then correlating it again with our scanning ion conductance topography. So this really then allows us to do this the topographical and volumetric distributions of the cytoskeleton components using these two modalities. And finally, in order to show the full power of the technique, we have transitioned to the live cell imaging, which has actually been done on the same setup. So here we can do in parallel scanning ion conductance microscopy and SOFI imaging of live cell, and we can take the nice time lapse images. I hope you can start to see the largest portion of the actin here, then the portion, the white field, and the combined SOFI second order and the SECAM topography in this lifetime time lapse from these different movies. So just to finalize, so we demonstrated the ability of the combined method for biological studies, and we hope that this will method will be further used to investigate open questions in membrane trafficking, cell migration, or infection. Ideally, I would also like to use this technique to study ion channels. So due to the electrical nature of the scanning ion-conductance microscopy measurements, we can also start to retrieve also surface charge information. So adding a whole new dimension to these measurements. So the ability also to additionally map the charges of the cell membrane, I think is particularly relevant for problems where we encounter clustering of the membrane proteins, membrane curvature, and other interesting systems. So with this, I would like to finalize my 3 stories, and I thank my whole laboratory that participated in the nanopore research. So here I mean without us, Andre, Mukesh, Martina. Then for the imaging part, Adrian and Christina Grusmeyer, and our collaborators in the aerolysin project, Cao Chan, Matteo Del Peraro, and the collaborators in France. And for the scanning ion-conductance microscopy collaborators, the developers of the high-speed microscope, Professor Fandner and Samuel. Thank you for your attention. I am happy to receive questions.
Okay, thank you, Aleksandra. So, very nice presentation. Let me check if there are questions. I mean, I have a lot of questions, but I will start with mine, and then if there are other questions, please write them on the chat. So, concerning the first part of your talk about sensing, if I correctly understand, you import the polymer electrophoretically because in essence, you are working with the DNA.
Yes.
I was wondering if you think to expand to other biopolymers or sugars that are less charged or neutral, and if in case you think that, I don't know, other approach like, I don't know, electrosmosis or a pressure field that can be used.
Yeah, so this is a great question. So we just recently built also a system where we can apply pressure in our flow cells. Still, I believe to move to something as a sugars, we still have to work a lot on also on the geometry of our pores. But I agree, pressure, addition of the pressure in the flow cell is very important because this can help deliver biomolecules which are not charged.
Okay, thank you. So it's okay.
And exploit the flow. Yes, exploit the flow. Yeah.
And another curiosity, instead of the fabrication part, I mean, I'm not an expert of this. You show that you can drill pore in your 2D materials of I mean less than 1 nanometer or around a nanometer. I was wondering if and how you can control the shape and which are the limitations.
Okay. So this is a great question. So we have very little control over the shape and also on the stability of the pore when it goes in the liquids. So we are working actually in order to stabilize these nanopores in the liquid so that they don't grow. By working in a oxygen-free, with trying to deplete oxygen from our buffers, and also in another line of research. So there are 2 lines. So one is which aims to stabilize the shape of the pore by attaching specific molecules. And for that, we started with the thiols. So the thiols binds to sulfur sulfur vacancies, and pore can be thought as as a place where a lot of such tiols can bind. So this is something that we are trying to do in order to stabilize the shape that we get. And another type is more electrochemical. Here, using the connected ribbon, as you see there, we exploit electrochemical reaction with dionysium salts, which can be also added any type of large type of chemicals, then bind covalently to the molybdenum disulfide, which is under potential. So these are two strategies that we are developing in order to stabilize the pores. In terms of the shapes, there is this one of the images showed little bit rectangular slits that we try to carve in aberration corrected microscope. So this is we are thinking that the advancement in the aberration corrected microscopy might also help us in that direction. But one thing that is also hindering the progress is stability of these pores in the solutions. And this is the first thing that I would like to tackle. And then the next thing would be the shape in a parallel, I hope.
Thank you. There is another question by Mamar Bubakcha, sorry for the pronunciation, is for sure wrong, and thank you for your great talk. What are the various obstacles you face in the pathway from ions to images with a microscope, and what is the solution to this problem, if
so I think what hindered really the scanning ion-conductance microscopy. So we see that atomic force microscopy area developed much more in terms of, if one would just compare the number of papers, the number of applications. And this comes, I think, from the fact that for the availability of the cantilevers, now we have really the super well developed companies that can provide well defined cantilevers with the nice probes. For scanning ion-conductance microscopy, this was not the case. Going smaller and having a company or having the technology that will produce reproducibly such probes limited democratization of this technique. So I believe the path is now open because we have a ways of having this glass capillaries very small, which then leads to the high resolution. So therefore interest from the people who are imaging biological systems, because they now approach the nanoscale. But still, let's say, I would say the company that would deliver us a field capillary, which would say, okay, this capillary gives non-rectifying ion current characteristics, and it's stable, is something that is very needed in the field.
Okay, thank you. There is time for, there are several other questions. I will select only one, short one for a short answer, then you can go on the chat and privately reply to the people. So the question is from Alberto Giacomello, and is, how is noise limiting this new strategy for storing data?
How is noise limiting? Okay. Yeah. So here comes the part of machine learning and also like classification of the molecules. So I went very quickly through this part, not showing really data for all position of the one along this construct where we have the 201, 20. So the one can move. Of course, as the one is closer to the A, our signals start to be less good compared to when it's in a really center position in corresponding to the two zeros. So again, here we started with one, let's say not one, but we tested several mutants. So the MD can help, but also I think the level of designing these polymers as well can help in increasing signal-to-noise ratio. So yeah, and of course now we have the machine learning, which is helping us deal with the noise. Having access to the lot of data and the training sets that we can do is helping. But for the practical applications, when we will of course need the long polymers, one needs to optimize as well the signal-to-noise ratio. Yeah.
Okay, thank you very much. There are another couple of questions on the chat that better that you answer privately. And
yeah, I would like to apologize for my camera and for the two technical glitches. So I hope this was not too annoying to the.
The talk was clear and it's
okay. Thank you.
Easy to follow, no problem. So thank you again, and